Hello class, this is chapter 3. We're going to get into a little bit of biochem, mostly talking about organic molecules. So there'll be a short little intro, that'll be this part, and then we'll have sections on carbohydrates, lipids, proteins, and nucleic acids. So a lot of this is about food, which is kind of fun. But anyway, let's get started. Alright, so when we say organic molecules, we're not using the same definition as a grocery store or maybe the FDA. In this class, when we say organic, we just mean that it has carbon and hydrogen, but especially carbon. There are very few molecules with carbon that are not organic, and those exceptions are the carbon-based gases like carbon monoxide, carbon dioxide, but the rest, like methane is CH4, it is organic. Organic molecules tend to be larger than inorganic molecules. Inorganic does not mean that it's not important to life. For example, water is inorganic, as well as oxygen gas. So we're just talking generally about size and then whether it's carbon-based or not. There are 4 basic classes of organic molecules that are in live bodies. These are carbohydrates, lipids, proteins, and nucleic acids. Most of these organic molecules are mostly carbon and hydrogen, and then they have some accessories. And we often call these accessories functional groups. And then I really want you to know 4 of these so that when we start looking at structures of these organic molecules, you know what you're looking at. So you'll often see a R. The R means literally anything. It's a placeholder for any structure. And then what you're really looking at is what is attached to that structure. And that'll give it some properties that are important in its reactivity. So the first group I want you to know is hydroxyl. And that is an OH group attached to something. It's common, any alcohol is something with an OH group, but it's common in monosaccharides, so carbohydrates, and it's one of the parts of a fatty acid, and it's also common in certain nucleic acids, but its property, it will add polarity to a structure and the possibility of a hydrogen bond. So that's hydroxyl. You find it mostly in carbohydrates, but in some other structures as well, and it adds polarity. Alright, the next one is the carboxyl group. And you could also consider it as the acid group, because it is the most common organic acid. And it's anything attached to a COOH with one double bonded O and single bonded OH. So it has a hydroxyl group as a part of it, and that's why it's called carboxyl. But it's this whole thing. So the COOH, it's polar, but it's also acidic, so it'll lose that hydrogen when it's in solution. It's common, it's the functional part of a fatty acid, and it's common in amino acids. So when we get to those, look for the carboxyl group. The third out of the four I want you to know is the amino group, and it's anything attached to NH2. So one nitrogen, two hydrogens. And it's polar, but it's also basic. So this is your acid, and the amino group is your base. And the amino group can also make hydrogen bonds. And you'll find those on amino acids and part of a nucleotide. The last one I want you to know is phosphate. So it's anything attached to a PO4, and it is very polar, and it's often pretty negatively charged. It'll lose sometimes both of its hydrogens in solution, and it's common in DNA and RNA most often, but you can also see it in some of the carbohydrate reactions when we get to metabolism. So those are the four I want you to know. Hydroxyl is polar, carboxyl is acidic, amino is basic, and phosphate is very polar and very ionic. You find hydroxyl in carbohydrates, among others. You find the acid group in fatty acids and amino acids. You find the amino group in amino acids and nucleotides. And you find the phosphate group in nucleotides and DNA and RNA. So those are the four functional groups I want you to know. When we talk about inorganic versus organic, again, inorganic, these things are small. They don't have carbon. A lot of the metal ions are inorganic, so they're really not attached to anything. It's just one atom. Salt, sodium, and chloride, those are both inorganic. When you think about organic, you should think about larger things, so vitamins, carbohydrates, proteins, fats, all that stuff. But the main basic difference is, does it have carbon or does it not? So why do we care about carbon so much? It's relatively small, and it has 4 valence electrons, which means it wants to share electrons to fill its valence. So it makes 4 single covalent bonds. It can also make double and triple bonds, but at its maximum number of covalent bonds, it can make 4. Also, it's going to want to make covalent bonds instead of ionic ones. So that's another important point. Because it's small, it's flexible on who it can partner with. So carbon can bind to nitrogen, hydrogen, oxygen, phosphorus, and sulfur. The carbon-carbon bond is very stable. It's not very reactive. It's non-polar, so it's very, very stable. And therefore we get long carbon chains or carbon rings that we can form. And on this structure, the chains can be very linear or they can branch. And the structure can have any of those functional groups on 2 slides ago. Any of those added to it. So again, the functional groups will be added to our carbon skeleton, and that will add some reactivity to our organic molecule. And with organic molecules, you can get compounds that have the same, essentially they'd be written the same formula. So C3, 1, 2, 3, 4, 5, 6H03. This one is also C3, 1, 2, 3, 4, 5, 6H or H6O3. So these have the same formula, but they have a different structure. When two molecules have the same formula but different structures, they're called isomers. So the main organic molecules in our cells are the biomolecules, and those are carbs, lipids, proteins, and nucleic acids. Most of these are long chains of repeating units. Now these chains can be cross-linked, so they can be connected to each other in a way that makes like a net. But these chains can also fold in on themselves and make a 3D structure. So you can get lots of variety. But each building block or each subunit is going to be called a monomer. And when you link them together and you have these long chains or nets or 3D model, we call it a polymer. So let's break down that word a little bit. So mono means one, poly means many, and mer just means part. So monomer is one part, polymer is many parts. And if you've ever made like a paperclip chain or a paperclip necklace, you've made a polymer, so you inherently understand this. If you've ever made a structure out of Legos, you've essentially made a polymer. But generally you have these subunits that are linked together. And when they're apart, they're called a monomer. And when we link them together, they're called a polymer. Lipids are not technical polymers, but we'll talk about them when we get there. Alright, so when we're taking single units, monomers, and we're transforming them into a chain, we use a certain reaction called dehydration synthesis reaction. So dehydration, when you add these units together, some water is freed up or removed. And hopefully they'll show it in a second. And so that's the dehydration part, and then the synthesis, synth means to make, and esis is just the process of. So synthesis is the process of making. We're making this polymer. The opposite direction, when we're going from a polymer to a monomer, we're breaking covalent bonds. The knife or the scissors that we use to break these bonds is actually water. So water goes back into the structure. And we call it hydrolysis. Hydro means water, and lysis means to break or to cut. So we're using water to break or cut these covalent bonds. We're going from polymer chains down to single units, to monomers. So here's two monomers, and we're about to add them together. When we add them together, we're going to remove water. We get half of it from one monomer and half of it from the other. And we'll make this covalent bond. So there's space between those monomers before the reaction, and after there is that covalent bond. So this is dehydration synthesis. Here's the dehydration part. Here's the synthesis. We made this bond. The opposite is hydrolysis. We're going to use water as a knife to break that bond. So here's the bond before it's broken. Water's going to go in and attack that bond, and then it'll break up our two monomers. There'll be a space there instead of a covalent bond. So know these two reactions, both dehydration synthesis, and this book may just call it dehydration, and the hydrolysis. We use dehydration to make polymers. This is a polymer. We're going to use the dehydration reaction to make polymers from our monomers. We're going to use hydrolysis to go the opposite way. We're going to break the polymer down into individual monomers. So there's a close-up of the hydrolysis. We're going to break this bond. Water will attack it. It'll separate the two monomers. Here's one half of our water, and here's the other half. And that is hydrolysis. Remember, lysis means to break or to burst, something along those lines. Typically, for both of these, both dehydration synthesis and hydrolysis, there's an enzyme that speeds up that reaction. It sort of drives it. And enzymes are typically proteins, so we'll talk about them more when we get to proteins. But they're also catalysts. So that word catalyst, it means that they're not consumed by the reaction. So they are not an ingredient in the reaction, and they're not irreversibly changed by the reaction. They will alter. Usually it's a shape change, but they aren't universally changed. And if you actually take biochem, you'll study these enzymatic reactions and what parts of the reaction the enzyme does, or the enzyme participates in, I should say. And it's pretty cool. Alright, so that's the end of our little introduction. Again, know the 4 basic groups of organic molecules, know those 4 functional groups I asked you to know, and then know those 2 reactions that we went over, and the definition polymer versus monomer.